This study focuses on the effect ketamine has on neurons in the cortex of mice and tries to find out how a dissociated state can occur. So, to get started, a dissociated state is loosely defined as a detachment from reality with varying degrees of intensity and duration. Basically, the brain can become detached from the environment and the body's sensory inputs while maintaining vivid, internally generated experiences. These can be caused by dissociative drugs, but can also occur naturally due to severe stress, trauma, and psychiatric disorders. The circuit and cellular mechanisms producing the dissociative state remain poorly understood, which is what this study aims to investigate. The dissociative drug being used in this experiment is ketamine. Ketamine is often found as a clear liquid that can be injected into the body, or as a white powder that can be consumed nasally. Ketamine has several clinical uses. It can be injected as an anesthetic and can also be used to relieve pain. It is considered safer than most other anesthetics and pain relief drugs due to the fact that it does not depress breathing or lower blood pressure, making them an essential medicine. However, the abuse of ketamine has increased in recent years. Its recreational use has grown in popularity due to the intoxication that occurs at high doses. Ketamine, however, is extremely powerful and can easily lead to overdosing. The main data used for analysis in this experiment came from electroencephalograms, or EEG. An EEG is a test that detects abnormalities in your brain waves or in the electrical activity of your brain. These charges are amplified and appear as a graph on a computer screen. While electrodes placed on the scalp are often used for humans, the mice in this experiment receive surgically implanted head-mounted electrodes to ensure that they stay in place and produce reliable data. The experiment was done completely on mice, around 2 to 3 months of age. First, a genetically encoded calcium indicator called GCAMP6F was used for calcium imaging of pyramidal cells and interneurons in the cortex. Mice were also injected with AAV1 to serve as a control for this series of experiments where interneurons were activated and pyramidal cells were imaged. The injections were performed in 1.5-month-old mice, who were subsequently prepared for head fixation and imaging after 2 weeks of AAV expression. Mice were put under anesthesia and were given head mounts, and after 3 days of rest, the experiments began. Each mouse was given 1 dose of ketamine per experimental condition. They could have received ketamines in amounts of 10, 50, 100, and 150 micrograms, or they could have received saline instead. All doses were delivered via a single injection. The neurons chosen were each examined for 2 minutes. Data was analyzed using several methods, the first being a behavioral analysis. Mice were assessed before and after an injection of ketamine or saline. They did several tests, including a marble burial test, where they saw how many marbles they could bury in 30 minutes. There was also a reactivity or limb withdrawal test to a puff of air, and an AR assay, where adhesive was placed on the snout of the mice, and the mice were given 150 seconds to remove it. Next, there was the EEG, which was explained in an earlier slide. Finally, there was 2 proton and vivo recordings. When the neurons fire action potentials, intracellular calcium levels rise, which can be detected using fluorescent molecules that bind to calcium, which is used for recording neuronal activity in an intact brain. In terms of the behavioral study, mice injected with the ketamine had reduced performance on all tests and did not respond to non-painful stimuli. They failed to bury many of the marbles, remove the adhesive, and many didn't withdraw their limb from the air puff, indicating impaired sensorimotor processing. It also reduced the EEG recordings of slow wave activity compared for wakefulness, but didn't seem to affect the high frequency oscillations. The first major discovery was that ketamine induces a switch in the active layer 2-3 neurons. Highly active neurons became less active or inactive under ketamine, whereas a subset of neurons that were inactive or weakly active during wakefulness became activated. Despite the activity switch, ketamine preserved or even increased the overall average spontaneous rate of layer 2-3 calcium across all neurons relative to saline controls. To determine whether this neuronal switch was restricted to layers 2 and 3, they imaged and analyzed the activity of neurons in layer 4, an input recipient layer, and layer 5, an output layer. Both layers 4 and 5 neurons also displayed a ketamine-induced rapid and persistent switch of neuronal activity, with highly active cells becoming less active and inactive or weakly active cells under wakefulness becoming activated. Next, the researchers wanted to see if this activity switch was found in more than just the primary somatosensory cortex. So they imaged and analyzed the neuron activity of layers 2 and 3 in other sensory regions, like the primary visual cortex, a neighboring motor region, the forelimb primary motor cortex, in two associated areas, the secondary motor cortex and the retrosplenial cortex, with ketamine at both doses in all image regions. Ketamine suppressed the activity of highly active neurons and activated inactive neurons. Researchers wanted to know if ketamine was causing the switch locally or systematically. Because the change occurs across the cortex, ketamine could be acting on either the subcortical nuclei or the thalamus. However, when injected locally, ketamine still caused the same effect on layer 2-3 neurons. Systematic ketamine delivery is still strongly correlated with the activity switch, and the experiments imply that both systematic and local ketamine affect similar neuronal populations, and that the ketamine-induced neuronal switch is reproducible in similar neuron populations. Simply put, ketamine can affect the entire cortex, but is also able to make changes in a small population of neurons. It seems that the inhibition of GABAergic interneurons is a big part of why the activity switch occurs. First, it seems that pyramidal activity switch requires both PV and SST suppression. These are both inhibitory interneurons. And it seems that both systematic and local doses of ketamine, but not saline injection, induce a generalized suppression of the spontaneous rate and peak response across all interneurons from baseline measures. This is in contrast to their neighboring pyramidal cells, which generally maintain their signals during transition. This was further supported by the fact that the active switch did not occur as frequently when the interneurons were left on. VIP neurons, or another interneuron, tested that may have an effect. However, the researchers were not able to say for certain. It also seems that the inhibition of NMDAR, or HCN channels, drive the switch of pyramidal activity. Ketamine suppresses NMDAR channels on active interneurons, as well as HCN channels on pyramidal neurons. Suppression of these channels using different methods seems to mimic the effect of ketamine. How the brain generates internal experiences disconnected from the surrounding world is still unclear. Doses of ketamine do cause a substantial reorganization of spontaneous neocortical activity, where previously active neurons become preferentially suppressed and a subset of previously silent neurons become activated. While this information still only gives us a novel understanding of the circuitry inducing these non-ordinary states of consciousness, it may explain how ketamine can disconnect sensory cues from cognitive processing. Evidence from this experiment argues ketamine's behavioral effects stem from cortical mechanisms. The divergence of cortical activity from thalamic inputs may explain why sensory experiences, such as hallucinations during ketamine-induced dissociation, are unrelated to sensory inputs from the environment. This study discovers many of ketamine's molecular interactions and is a good first step to finding out how a dissociated state happens. Ketamine is extremely important medicine and is used very frequently for clinical purposes, so understanding how it affects our brains will be very helpful in the future. Overall, this was a very interesting experiment that was able to continue building on itself with each new discovery. There are, of course, complications when it comes to comparing research between species. A dissociated state is a subjective experience, and as such, it is difficult to experimentally characterize, especially in rodents. The way ketamine was administered in this experiment is different to how most humans receive it as well. The researchers made sure to address these complications, however. The paper itself was also well written, however, it would have been nice for them to spread the figures out throughout the results to better illustrate rather than just clumping them all together.